Oh yeah, get record our sessions. So that's pretty cool. And then, you can like go over them if you want. So, okay. Yeah, bioinformatics, basically you use like computational methods to figure out the meaning behind the information contained in organisms based on their, genomes. So there's GenBank, right? And that's it's an online repository of sequence data. You can just access it yourself. There's also RefSec, which is basically an annotated version of a species' genome. So that's what we basically use when we do any type of bioinformatics like research. And like, so that's why I was talking about like a genome library and a cDNA library, right? Like if you wanted to do research, right? Like I had to use like an entire human genome, which is like a 40 gigabyte file. And then to actually do bioinformatics with something that's that big of a file, you need like a really powerful machine, like, and you just see how that's super like inefficient, right? Whereas if I just used a cDNA library, which just takes the exons, right, which is just how much percent of the genome?
2%.
Correct. So it'll just be like,
to 2.
yeah, so it won't be a 40 gig file, it'll be like 4 gigs. No, even less, like 0.4, like
Should be like a gig.
400 megabytes or something. Yeah. So you see how, so that's like the reasoning behind why we use a cDNA library instead of an entire genome.
Yeah.
Okay. All right. Yeah, this is what we use also. BLAST stands for Basic Local Alignment Search Tool. Allows users to find a nucleotide sequence or an amino acid sequence that's related to any given sequence. So let's say we looked for this whatever sequence up there, like in the query, and then you'd return like matches that are similar to that. So yeah, that's basically what you use like on a computer. Yeah. So okay, and I knew that they were talking about hemoglobin. Wait, go up. I mean, yeah, but underneath that. Yeah. Next one. The side after that. Yeah. So okay. Yeah, that one. So basically, like, so before when we were talking about alpha and beta, I knew that they were going to talk about hemoglobin. It's a perfect example. So, all right, we're going to use hemoglobin as an example. So you can move on to the next slide. So there we go. So hemoglobin has basically alpha globin group subunit and beta subunit. So remember like how like proteins have like primary structure, secondary, tertiary, and quaternary. So some proteins don't have a quaternary structure. Quaternary structure is the interaction of subunits. So myoglobin, for example, which is what like is the muscle equivalent of hemoglobin, right? It's just a tertiary structure, meaning that it doesn't have an alpha and beta subunit. It just has one subunit, which I guess is just a unit. It won't be called a subunit then. Hemoglobin, however, has a quaternary structure because it has multiple subunits. So it has 2 alpha subunits and 2 beta subunits, right? And then there's a polypeptide chain that surrounds the heme group, which you see in like, those like kind of like disc-shaped things. That's the heme group that basically, it's like the thing with like iron in it that binds to like the oxygen itself and releases it. So yeah, that's basically what hemoglobin looks like. Good to know that. Comes up all the time. All right. Now, okay, and then they're saying so what we saw before, that's an adult hemoglobin. So it changes during development because okay, embryonic, so you have, I forgot what that Greek letter was called, but let's call it Z. And then I think that's epsilon. Those are embryonic. I think, yeah, I might be wrong. And then fetal, that's gamma. Hemoglobins, they bind more tightly to oxygen to facilitate the transfer of oxygen from the mother to the embryo or the fetus. And that makes sense, right? Like the version of because like how else would a baby, like, you know, like a fetus or whatever, get oxygen, right? They don't have their own lungs, right?
Yeah.
And so it basically means that their hemoglobin has a stronger binding affinity for oxygen than adult hemoglobin does. So when our blood goes to the fetus, our hemoglobin gives up its oxygen to the baby's hemoglobin. So you see, yeah, so you see how like the so this is a graph of like whatever weeks of development, right? And the proportion of these types of hemoglobin. So you see how the embryonic one, from 0 weeks of development, like it's like 30% of it, to like 6 weeks, it's 0, right?
Yeah.
Yeah. And then the fetal one starts to increase as the embryonic one decreases, right? And it increases from like around 6 weeks, right? To around like 12 weeks. And then it starts to go down, right?
Yeah.
As it starts to go down, the adult beta subunit, right, starts to increase. So that's why you see that inversion there between the gamma and the beta. As the, you know, fetal hemoglobin starts to decrease, the adult hemoglobin subunit, the beta, starts to increase.
Yeah, and it keeps increasing all the way from birth.
Yep. And then, you know, to development. And then it seems like, you know, alpha kind of like starts to increase.
Pretty much stays the same.
Yeah, around like 6, like, or whatever, 4 weeks, and then it stays relatively constant.
Yeah.
So yeah, that's what, you know, that graph, that chart means.
Got it.
All right. The order of the hemoglobin genes on the chromosome reflects the timing of the expression. So okay, chromosomal organization, they're genes that are in order on the chromosome. So, you know, they're in the same direction. LCR means it's a locus control region. It just basically it controls the sequential expression. Sequential means the order of it, like the order matters, like in the sequence matters. So the locus control region controls the sequential expression of genes at a locus. So if we look at that example of a normal beta cluster, right, you see there's like the epsilon 1, the G gamma, the A gamma, the like phi beta 1, the delta, the beta and stuff. They're like in that order, and that's determined by the LCR. It determines that order. Like it has to be, and that order matters in order for it to make the right version of the hemoglobin.
Got it.
Okay. So like that's why, you know, the epsilon 1 comes before like the adult ones. But anyway, yeah.
Got it. I got it.
Hereditary persistence of fetal hemoglobin. So there's a rare condition where you continue to express the fetal hemoglobin level, right? So like compared to that normal beta cluster up top, if you had hereditary persistence of fetal hemoglobin, you won't be making, so you'll have a deletion of the delta and the beta genes, right? And it says it should be lethal, but let's say that, you know, it's not, so you still survive. The gamma genes continue to be expressed, and that results in a near normal level of health. So from the normal cluster to the hereditary persistence of fetal hemoglobin cluster, that you see that red X, that thing, like it stops, the LCR gets messed up and it stops making the delta and beta genes, right? But it continues to make, you know, everything else. And by the way, you see how it makes that purple gene, it doesn't affect that.
Yeah.
Yeah, that purple gene is still there, but it is not affected by the LCR. Okay.
Yeah.
Okay. All right. So these are mutations that cause globin-related disease. So there's hemolytic anemias, which, you know, is from a change in the amino acid sequence of alpha or beta globin chain, causes destruction of red blood cells. You know, hemolytic. You know what it means. An example is sickle cell anemia. And there are thalassemias, where the mutations reduce or eliminate production of one of the two globin polypeptides, and there's a range of phenotypes that is shown in. So here's an example, normal, right? Example of thalassemias. So like normal ones. An example of thalassemia is associated with alpha globin deletion. So okay, normal, right? Alpha chain production 100%, right? You know, number of functional alpha genes is 4. So, and then there's a silent carrier where it has 3 alpha functional genes and produces, you know, 3/4 of it. There's heterozygous alpha thalassemia, which is basically mild anemia, where only half of the alpha chains are produced. Then there's basically, you know, homozygous or not homozygous in between there, where it's moderately severe anemia, only 1/4 is produced. And then if you're homozygous, it's completely lethal. Yeah, nothing, no alpha chains are produced whatsoever, and you die.
Got it.
So yeah, severe here, loss of the gene expression. So that LCR is deleted, and the LCR basically determines the sequential expression of those genes. Bless you. And so if it's deleted, you're not going to have those genes expressed.
Uh-huh. Got it. Well, that's the end of that slide. The end of those slides.
All right. So tell me again, what's alternative splicing?
Alternative splicing.
Mm-hmm.
So what do you call it? It's just like, well, also what I have like written here. So like one gene can produce like a whole bunch of different proteins. So if it has like different, let's say, numbers, so that example, like 1,
Of
2, 3,
exons,
4
right?
can create,
Mhm.
yeah, of exons. Like 1, 2, 3, 4 can create like something for the kidney, and then like 1, 2, 1 can
Exactly.
produce something for the liver,
Good.
and then
Good.
like 1, 1, 4 can produce something for, like, you know, the lung or whatever.
Good. Awesome.
So yeah.
Mm-hmm.
Okay, exam 2 is on chapter 7 through 12, so we won't go over any more PowerPoints just for today.
Okay.
Just so we can do that, and then we'll just like hyper go through the rest.
Okay.
And stuff like within next week and whatever. In the next coming weeks. So let's see. Right, yeah. Exam 2 is chapter 7 through 12. We've done that. So then what else? Yeah, we just went over chapter 11. So it's 13, 16, 24 that we've done. That we still gotta do. Then we'll get 18 on Monday. Yeah. Then we can just go through those. Okay. What else? What else? What else? Oh, my complex ion report.
Mm-hmm.
Okay, so I'm gonna put this my port on the left with data, whatever,
Okay.
and I'm gonna put what it says.
Nothing. Fix your spelling of burette up there, but I think you know that.
Oh yeah.
It's a
Um.
TTE, but okay. Moles, yeah. You just take the molarity and you multiply it by the volume. Oh man, that's funny. They spell it wrong in the thing.
Yeah.
All right, whatever. Anyway, so okay, what
Okay.
do you need help with here?
So hold on. Let's see what this girl did because she sent me like how she did her part, whatever. Oh, okay. So you don't even put part 1 in the lab. I see. All right. All right. So there's no part 1 in the lab because you didn't even. There's nothing to make. Got it. Okay. Let me just fix it out this for the iPad. Okay. Okay, so I guess for part 2, I need to read content over here. What it says on the left. I can do my best to zoom in. Hold on. I know you're seeing on a small screen. How's that? Is that better?
I could have I could have read it before. I could see
Oh,
you could go back to the way it was before. Then I could see more of it.
yeah.
Yeah,
Okay, so
that's good.
yeah, part 2. What is it asking me to do in these calculations over here?
So you have to calculate the moles. So basically, you know, you take basically wait. Okay. So you have the volume, I guess, in milliliters. And then
Okay.
let's see.
Okay.
You have to find the moles, okay? So mass, well, let's wait, moles of ammonia per gram. Wait, okay. So moles of HCl, moles of NaOH, and then moles of NH3 is the moles of HCl minus the moles of NaOH. Okay, so what's that mass pertain to?
Which one?
Like trial 1 and trial 2, like up top, like 0.209 grams. What's that for?
Oh yeah, it's just like I guess the mass of whatever the solution was at the end of it. Because they were performing an indirect titration, so I'm guessing you're supposed to do it on like a tiered beaker and stuff.
Yeah.
You're supposed to see what the mass was at the end and whatever. It was just part of the data set they gave us, but I'm pretty sure it doesn't have anything to do with any of
Then
the calculations here from what I see.
so we also need molarity. So you know the molarity, is that what you have on the bottom there in part 3?
Yeah, the molarity of all substances used I have
Okay.
down here.
So this is what you
It's
do.
after part 3. Yeah.
You take so you subtract the final HCl from the initial.
Yeah.
You convert that into liters and you multiply it by the molarity to find the moles.
Okay, so minus the final by the initial.
Yeah, so 23.89 minus 3.95.
And then I got 19.94 and I move it 3 spots to the left.
Yeah, just divide it by 1,000.
So I get 0.01994.
And then multiply it by molarity. So multiply it by 0.3034.
And I get 0.0604.
Yeah, so write that down as moles of HCl.
Okay. For HCl for trial 1, I guess. Yeah.
Yeah.
I'm just gonna put, it should be 0.006, right?
Yeah, that's what you got, right?
Yeah, even though there's 0.4 and stuff, it should just end at that 0.
Okay,
Cool.
now do the same thing for NaOH. So 29.67 minus 4.94. I mean 4.91.
So I get 0.0028.
Oh, you divided by 1,000?
Yeah, I already did that. And then times it by the molarity.
Oh, you multiplied it by 0.1169?
Yeah.
Oh yeah, yeah. So good job. So yeah, that should be moles of NaOH in trial 1.
Okay. And then there's zeros, it's 0.00289, so I should just do 0.003.
Just use like the most whatever accurate ones, and then we can round it at the end, I guess. Because there's
So
one last step after this.
I'll put 0.0028.
Okay, and then moles of the ammonia is moles of HCl minus the moles of NaOH.
Whoa, what happened? Sorry.
Yeah, so now the moles of NH3 ammonia, right? That's going to be the moles of the HCl subtract minus the moles of the NaOH.
So 0.006 minus 0.0028.
Correct.
So then it's moles of NH3 for tile 1 will be okay. Trial 1, okay. And I get 0.0032.
Okay.
Do I put anything at the end? Be mole?
You don't have to,
Like
since
that?
you already said moles, but
Oh, okay.
But now what you do is you take that mass, so you get what you got for moles of NH3, and you divide it by the mass that you had for the sample. So you divide it by 0.209.
So take this and divide it by 0.209.
Yeah.
Okay.
And that'll give you the number of moles of ammonia
Grams per mole, right?
What's up?
Is that grams per moles or just moles of NH3 divided by the grams?
Yeah, so it'll be moles per gram. So basically, once you do that calculation, you're going to have the number of moles of ammonia per gram of the sample that you had.
Okay.
Does that make sense?
Yeah. So what should I put for the next thing?
You can just say
Moles per gram.
Oh yeah, say moles of ammonia or whatever, moles of NH3 per gram of sample. Yeah.
And then I got 0.01531. So I think I'll just go 0.0153.
Okay.
Okay. So that's it for the first one, right? And then after we get the both trials, we're supposed to average out the moles per gram.
Okay.
I guess that's what it says.
Mhm.
Average the results from both trials.
Yep.
Okay. So now for this one, we do.
For part 3?
Yeah. No, for trial 2. No,
Yeah.
it's the same thing, right?
Yeah.
33.87 minus 23.89 divided by 1,000. Then after we divide it by 1,000, we're supposed to
multiply by molarity.
by the molarity.
Mhm.
Okay. And we're doing this HCl. So it's 0.3034. It's like it's 0.0060 again. So 0.006. And now we do the same thing over here. So 40.45 minus 13.68. Divided by 1,000 times 0.1169. So 0.0031 is what I get here.
Okay.
So 0.0031. Yeah, 312 or whatever, but I'll just go 31. Whatever. So now I just minus both of these, right? 0.006 minus 0.0031.
Mm-hmm.
I get 0.0029. And now we take the moles of NH3 and we divide it by what?
The mass.
The mass.
So 0.196.
And I get this is here 0.0147 or 148.
Yeah.
Point 0.0148. Okay.
Yeah, and then you average those out. So you average out, so 0.0153 and 0.0148.
Okay. Sorry. Say it again.
Yeah, so now you just average out that, moles of NH3 per gram of sample. So, 0.0153 and 0.0148. Yeah, just average that.
Average of
Average moles of any, yeah.
from both trials. And then to take the average of that, I just add them and divide it by 2, right?
Mm-hmm.
So I get 0.01505. So 0.150. That's how I'll do it.
It'll be, I guess, closer to 0.151.
Yeah.
But yeah.
Okay. So then that's the result of part 2. Oh, sorry.
Bless you. Yeah. Now, bless you again. So now we're gonna be doing the same thing, but we're gonna find the nickel content. Okay. So basically, now we're just doing the thing with EDTA.
Okay.
So basically, take the V final of EDTA subtracted by the V initial, divide by 1,000 to get it into liters, multiply it by the molarity of EDTA to get the moles of EDTA.
So moles of EDTA.
Mm-hmm.
Trial 1, right?
Mm-hmm.
That's all we'll need, right? And then it'll be moles of Ni2 plus, right?
Mm-hmm. Yeah, that'll be equivalent to the moles of EDTA, but yeah.
Okay. Well, I can explain that in the discussion, maybe. Okay. 2. Okay. All right, so moles of EDTA trial 1. So what I do here is 25.03
Yeah, you know, you could even go, so like go to that next line where you
Mhm.
wrote moles of moles of Na, like the nickel.
Mhm.
Just have it equal. Yeah, like go to the beginning of that, press backspace twice, and then space equals space. Yeah, that'll kind of make it easier because you don't have to write it again.
Oh, okay.
Yeah, but it'll still be trial one. But yeah.
Yeah. Got it. Okay. And do the same thing for trial 2. And then I just put the answer below, right?
Well, then yeah, for below, no, you can put the answer. I guess you wherever you want. You could put it up top, but the next thing. So like, yeah, first we'll do this, and then I'll tell you what to do next.
Okay. So the B final minus initial, so I get 20.22,
Yeah, divide by 1,000,
1,000. Okay, 0.02022.
and then multiply it by molarity of EDTA, which is 0.05104.
0.001032.
Yeah, so that'll be that. So you can do like colon and then yeah.
So 0.00103, I guess.
Mm-hmm.
Yeah, because that's 101 makes sense. Or should I just 0.01?
No, it's good like that.
Okay.
I don't know like whatever how it's graded with the numbers, but let's put
I don't
like
know. To me, there can't be sig figs with that right now.
yeah,
That
like
wouldn't make sense.
yeah. Let's start off by putting as many as possible, and then if you need less, you can kind of edit that part.
Okay.
Yeah, and then do the same thing with the trial 2 data. Oh, unless you wanted to do the next step for trial 1, however you want to do it.
Oh yeah, let's do that.
Okay, so yeah, the next step basically is going to be moles of nickel per gram of sample. Yeah, kind of like that.
Okay.
Yeah, so now what you do is you take your number of moles and you divide it by the gram, the mass of that sample. So 0.00103 divided by 0.344.
Wait, what? Sorry.
Yeah,
Oh, the mass. Sorry. Got it.
correct.
And then we divide it, right?
What's that? Yeah,
Divide it.
yeah, you take the moles and you divide it by the mass. So 0.00103.
0.000299.
Yeah. Yep.
And then we have to average the results from both of the trials.
Mhm.
And then after that, we have to take the average
Yep.
from up there.
Yeah.
Okay.
Just like you did for. Oh, sorry. So yeah, we're gonna.
Yeah, we average the results from both trials here, and then
Yep.
we take both averages and take the average of it.
Yeah. To find
Okay.
the average moles of ammonia per moles of the per moles of nickel.
All right. Well, we'll figure that in a minute. So
Mhm.
20.28 minus 1.58 is that divided by 1,000 times it by the molarity.
Mhm.
0.051, right? Wait, did I do something wrong here? I did 20.28 minus 1.58, right?
Yeah.
And then from that answer, I divide it by 1,000.
Yep.
And then I times it by 0.05104.
Yeah.
Okay. So I get 9.54 here.
Wait. That shouldn't be it.
Let me do it again.
Yeah, do it in a stepwise fashion, because if you're doing it all at once, there might be issues with parentheses in your calculator.
Yeah, no, I do it at stepwise. So hold
Okay.
on. 20.28 minus 1.58. Answer 18.7. Now divided by 1,000, right?
Yeah.
Okay. And now I take that answer
Mm-hmm.
and I times it by
Mm-hmm.
0.05104.
Yeah.
Let's look at the same answer.
So you should have gotten like 0.00 or like 0.018 or something for the volume, right?
Yeah, all right.
Is that what you get?
Um, for the volume, I get. Wait, the volume is what?
So you said it was like 18 point something milliliters.
Yeah,
So
18.7, and then when we convert into liters, it's 0.0187.
so if it's 0.0187 times 0.05, no, are you sure it's 9 point something, or is there an e at the end?
There's an e at the end.
Yeah, so it's an e to the negative exponent, right?
Yeah.
Yeah, so that's your answer. Whatever you get,
Oh,
yeah.
so okay. So the moles then is what?
So you're getting the right answer. It's just in scientific notation.
Is it written like this?
So yeah. So wait. So what does it say on the calculator? It says
-4.
okay.
9.544448
Good.
to -4.
Yeah. So you move it to the left 4 times, and yep, that's it. Good.
It's 3 zeros, right?
Yeah.
Okay. Got it. Okay. So now the moles per the gram of the sample is done.
Yeah. So this is where you take your answer and you divide it by the mass of the sample. So you divide it by 0.316.
So I take that answer
Mm-hmm.
that I have.
Yep.
So it's 0.000954,
Yep.
and divide it.
Yeah, by
By
0.316.
So I get 0.0030, so 0.003.
Yep.
Okay.
Yep. And then you average those out.
I average which of the results? The
Moles of nickel per gram of sample. Yeah, just yeah. Yeah. So 0.00299 plus 0.003, divided by 2, it's going to be like 0.00 like 2985.
Yeah, 0.002995.
Yeah.
So I'll just put 2995, so 299, I guess.
Yeah, it'll whatever round to like 0.003, I guess, but yeah.
Yeah, I might as well just do that. Yeah.
Oh yeah, and then the last step, basically, you take your average moles of ammonia per gram of sample from both trials, and you divide it by the average moles of nickel per gram of sample from both trials, and you'll get basically the moles, basically like a mole ratio, right, of ammonia per whatever moles of nickel.
So we take the
Yeah.
average from both, right?
So this is just going to be, oh, I see what you're doing. So yeah, I guess you could do it that way. We're basically finding the mole ratio so we can figure out the chemical formula of the complex, if that makes sense.
Yeah. Okay. So after dividing both of the averages,
Yeah, so do 0.0151 divided by 0.003.
I get 5.03 repeating.
Yeah. So like, let's say that thing that you wrote up top, let's make that equal to X.
That I wrote here.
Yeah. Like maybe like
Okay.
space or whatever equals X. Yeah, you're gonna have, I guess, play with the formatting to make it like in between or whatever. But yeah, you could just say, it's equal to X and then put another equal after that and put your answer down.
You know what? I got an easier way of doing this.
Yeah, I think you could do it that way. Yeah, that's better.
Equals X. Right?
Mm-hmm.
Like that.
Yeah. And then, put your answer there. What did you get?
Huh?
Yeah. What did you get for X?
5.03 repeating.
Okay. So yeah, you can put 5.
Just 5?
Mm-hmm. Yeah. And now, see that chemical formula?
Yeah.
We're going to write it down and we're going to do, see how it's 6 minus X?
Yeah.
And then X at the end. So now we know the chemical formula. So it's basically, Ni, parentheses H2O, 5, 1, because it's 6 minus 5, right? So 1. And then 5 ammonias.
Yeah.
So that's just a way so you started out with some compound, and through titration, you basically figured out the exact chemical formula of that compound.
Okay.
It's pretty cool.
So 6 minus X. So now we can just put equals, right?
Yeah, or maybe like a equal with an arrow, like a, arrow after it or whatever. I don't know. Yeah, oh yeah. And then you'd write that again, and now you know the formula, which is just, like, yeah. Since, we don't write the 1, you could just leave that, and then at the end of the ammonia, you can put the 5. Yeah. So that should be the formula.
Formula of complex ion.
Yeah. Chemical formula, yeah.
Got it.
Mm-hmm.
Nice. Okay, so now I guess for these questions, right? These discussion questions. What the fuck?
See, if the sample of the complex ion that you prepared had not thoroughly dried, but contained traces of water or ethanol, would this make your determination of the moles per gram of ammonia and moles per gram of nickel too high, too low, or would it not affect your results at all? Would this make your determination of X too high, too low, or not affect your okay, so let's say that there's going to be some extra water, right? And since nickel is complex to the water, you're going to have, kind of like an excess amount of that.
Yeah.
And so, okay, let me think about how it's going to actually affect things. So okay, let me ask you this. So is this how like the experiment was? Like, so they gave you some type of solid sample, right? It was a solid.
Well, we didn't do it in class, so we wouldn't know.
um, so like go to, yeah, on that left, on the document or whatever, on the left, go up to the part 1. Let me just see what's going. So okay. So let's see. Okay, so yeah. So weigh out 3.5 grams of this thing, right? In a 50 ml beaker, and then you add NH3 to mix it. Okay. Now, then, so you do start with a solid. Okay. Now, oh wait. What's above the procedure? Let me see, because I don't really know what the overall thing purpose of the lab is. So if you go all the way to the top, let me see. Okay. So all right, here we go. So okay. In aqueous solutions, the nickel cation forms a complex with 6 H2O ligands. All right. When this complex ion is mixed with ammonia, a reaction occurs where some of the H2O ligands are displaced with ammonia ligands. But how many are displaced by ammonia? So let's see. You mix the solid nickel 2 sulfate with and aqueous ammonia to synthesize a compound containing a complex ion whose formula is, what we basically are trying to figure out. Now we have to figure out, okay, so it acts as a base in aqueous solution. Okay. Okay. All right. So if you do the titration, they both appear blue, so you can't really tell when the titration's finished. So then we use Mirexide as an indicator. Okay, so now let's say that, okay, so, first we have the thing. So, okay, let's see. Sample not thoroughly dried, have traces of ethanol.
We write this down.
No, I'm just reading that and trying to figure this out. So basically, let's see. Okay, now can you like scroll down to the procedure? Okay, so let's see. So, all right, we weigh this out, and then we add the NH3, make sure all the solid is dissolved. Okay, then measure out some ethanol. Okay, and then mix it. That should cause the nickel ammonia complex to precipitate out of the solution. Okay. I think, all right, I think I understand. So basically, so you have like this solid amount, this solid like thing, right, of nickel, whatever, sulfate, and you add some, so that first step, when you add the ammonia, it's gonna replace the the 6 whatever H2Os with ammonia. Now, after that, you mix it with ethanol to cause that. So, okay, you start off with the solid version of the nickel sulfate, right? And you mix it with ammonia to make, the, complex that we found, right? And then since we mix it with ammonia, it's like aqueous or whatever. We throw in ethanol to make it precipitate back into a solid. Make sense?
Okay.
So now they're saying, what if that solid that you make in that second step of part 1 wasn't thoroughly dried, or if it had still some traces of water or ethanol, right? So that means that basically not enough of it has precipitated, not enough of it, has we didn't get all of it. So some of it is still in the solution. So that should mean that we should have less of the number of moles of ammonia per gram. And it should mean that, let's see. So yeah, it'll definitely make the number of ammonia less. And let me see how it would affect the number of nickel. Oh yeah. See that up there? So like the aqueous solutions, that cation forms a complex ion with 6 H2O ligands, right? Then we mix it with ammonia, right? Where some of the H2O ligands become displaced with ammonia ligands, right?
Yeah.
Now, how many H2O ligands are displaced by ammonia? So that's what we're trying to figure out, right? And in that step of, mixing the ethanol, you get the amount, the complex of the ammonia version of the nickel. So okay. Now, if there's still some water there, not all of the complex has been turned into that precipitate. So some of the complex is still going to exist as the nickel is. Some of the complex is still going to have. So okay, how can I explain this? So if it's not thoroughly dried, right, more of the nickel cations are going to be in a complex ion with the H2O ligands than ammonia. We want it to be all ammonia, but since we haven't thoroughly dried it, some water is still there, so less ammonia is going to be made. So it's going to be less of that complex with ammonia made.
Okay.
So okay. So basically what'll happen is less. So okay, you can go back to the discussion. So yeah, they're saying that if it's not thoroughly dried, so okay, and since we have to explain, right? So let's see. So okay, say that it would make our it would make our determination of the moles of ammonia per gram smaller.
Okay. Wait. Say it again.
Yeah. So say it would make. See here, I'll write it in the chat. It would make our measurement of, or rather, whatever. Let's see, our determination of moles of NH3 per gram of sample too low. And for explanation, I'm going to write this. Since ammonia begins displace the H begins
All right.
Let me try to think. All right. So each nickel will have 6 H2O ligands complex with it, whereas if it was ammonia, it would be 5 ammonia ligands. So we don't have enough ammonia. We don't have enough ammonia, so more of it's going to be with complex with 6 H2O. So since you need more H2O per I think it'll make it too low also.
Okay.
Okay, so the amount of nickel per gram of whatever sample would be let's see. I think, all right, I'm not sure of this, but would be too low as well, since not enough would have precipitated out to form the new complex. That's what I think.
The amount of nickel per gram of sample. Yeah, I think this is good enough because it's how much they usually ask for.
Okay.
As well, since not enough there. Perfect.
All right, the next one. Why can ammonia function as a ligand with the nickel cation, while ammonium cannot function with the nickel cation? Use structures to illustrate your answers. Okay. I guess like.
Oh yeah, let me see how this scroll
Draw
did
it.
it here. Actually, this illustration. I don't know why she did that.
Yeah. So see that? Okay. So see
Do we use
that?
the same exact ones?
What's that?
That's pretty much gonna be. Do we use the same exact ones?
Yeah.
Would that be
So,
part of the answer?
oh wait. So that's the answer right there, kind of.
Well, yeah, it's the other girl. Just send
Yeah,
me
that's pretty much
hers
it.
because I was lost.
Okay.
All right, well, I'll put the images here, but we'll make our own answer based on that. Okay. Oh, okay, then what you just typed as the answer here?
Yeah. Since these structures are from a different person's lab report, you should maybe use a different, image of the structure,
Okay.
just to be on the safe side.
Yeah, so that pretty much makes up the answer. Okay, so we'll do that. Okay, what do I search it up as?
Here. Oh, okay. So do a Google image search, I guess, for ammonia as a ligand. Let's see.
Here.
Keep scrolling. Oh, you know what? Write ammonia as a ligand with nickel. Yep. You can go to the maybe that one underneath it.
Huh?
Wait a sec. We got 5 as our answer, right?
Yeah.
Okay, whatever. Oh wait, 6 minus x.
Is 1.
Yeah. All right, you know what? Just look up whatever, a regular structure of ammonia and just put that down, I guess. Yeah, that one's good.
This one?
The one above that.
This one?
Yeah. Yeah, and then that one for ammonium is already there.
This one?
No, on the Google image.
Yeah.
See, like right, yeah. You could, I guess, use that. Yeah, these are better images anyway, since they have the geometry as well. Cool. All right, let's see. Copy the structure of EDTA 4 minus from your textbook and circle all 6 atoms that act as Lewis bases. Why does EDTA 4 minus cease to function as a chelating agent if acid is added to the solution?
So she did this.
Yeah. Look up your own thing though.
Okay. So what can I search it up then, please?
Just EDTA structure, like Google image search it. Yeah. So you could use,
This one?
um,
Or this one?
I like the one like on the top left.
This one?
Yeah.
This one?
Mm-hmm. Because that's how I would draw it. So now the atoms that act as Lewis bases. So Lewis base is basically a electron pair donor. So you're just going to circle the things.
What am I circling?
So it acts as a Lewis base, which means that it's an electron pair donor. So you're going to circle the things that have, basically electron pairs. So you can circle basically like the oxygens with the lone pairs and the ammonias as well. You could just circle whatever
Like
that girl circled in her thing, but like yourself.
So circle all the O's. Is what she did.
Yeah, the O's like at the end, not those O's.
Not these?
No.
Oh, these.
Yeah. Oh crap. Wait, sorry. We're using all right, we're gonna use a different one. This is actually EDTA, but they want EDTA 4 minus, which means all those OH groups lose those H's. So we need it to lose the H's. So let's look for that's like yeah, just scroll on the Google image search, scroll down. Keep keep scrolling. Okay. Um there's a couple of these. Uh okay. That
Here?
the one on the right. Yeah, that'll
This
work.
one?
Wait, damn it. That one has here, let me see what I can find.
You want me to put in EDT 4 minus?
Oh yeah, you can do that.
Okay.
Um
All right, so we can actually use the same thing, no?
let's just, there's one down there. I see. Go down a bit. Keep going. Like hold on. I'll just send it to you here. Copy link address and here in the chat. All right. I sent you the link in the chat. Did you open it?
Yeah. Hold on. Okay, let me see. Whoa. Okay.
Yeah, that's it right there. Mm-hmm.
This one?
Yeah. And then yeah, circle, the O minuses. Yeah.
And what else did I circle?
What did that girl circle? Did she circle anything else?
Circle the
Yeah. Oh,
end.
yeah. And then there's lone pairs on the nitrogens as well. Yeah, that one, and the other one.
Okay.
So, yeah, do you have to explain that, or
From what it looks like over here, no.
what did? Yeah, let me see that question. So, copy the structure, circle them. Okay, yeah, no. Why does it? Okay, so we have to answer that. Why does it cease to function when acid is added? Okay, so EDTA functions as a Lewis base. If so, wait, I'll just say EDTA 4 minus is deprotonated, and it's negatively charged. You know, negatively charged. All right, wait. EDTA 4 minus is deprotonated and acts as a chelating agent due to its negatively charged oxygens and lone pairs on its nitrogen. However, if an acid is added to solution, these EDTA would be protonated and no longer have negatively charged oxygens. Therefore, it would no longer act to chelate. Yeah, maybe write like nitrogen atoms, I guess, to the nitrogens, but yeah. Wait. So you kind of missed something that I sent here. In between where I say deprotonated, I wrote, however,
Uh-huh.
if an acid is added. So after that first sentence,
EDTA is deprotonated.
Yeah, that first sentence is fine. But after that first sentence, I wrote, however, if an acid is added to solution.
Oh.
Yeah. Make that period after the solution a comma.
However, if an acid is not a solution, EDTA will be protonated and no longer have a negatively charged oxygen. Period. Therefore, it would no longer act as a chelating agent.
Yeah. Yeah, I just put that asterisk to mean that I corrected myself.
Into a period, yeah.
So like that first picture that we used was the protonated form.
Got it.
Yeah. Also, in general, like you should justify all your paragraphs with like that the middle justification. Like you know what I'm talking about? Like all right, like if you like highlight like whatever, like everything, and then
Uh-huh.
go to like hit like control whatever A, and then go to where is it on the thing? So you know like where the justification is on your on the formatting, the one that's like up on your taskbar, the one that so okay, yeah, go keep going to the right, keep going there, like left a little bit, left, yeah, hit that, yeah. See how it's like all nice? I mean, you're gonna change some of that stuff, but it just made the words on the left and right like a block. You see,
Yeah.
looks nicer, right?
Yeah.
Yep. Yeah, you'll just have to fix some of those things. Yeah.
Fix. There we go. Good. Okay. That's good.
What do you write for conclusion?
Conclusion, I'll just write that like the experiment was successful in that, we were able to find, it's short, it's like 2-3 sentences. And then the abstract, and then the discussion, I'll just have to, all I
Did
gotta
that
do
girl
is,
get the same complex formula?
oh, and nobody got the same complex because
I
they
see.
gave us all different sets of data.
Um, okay. I wonder what what she got though.
Okay.
Yeah, yeah. Can you check what she got? I'm just curious. Oh, you know what? It makes sense now. But I just want to see anyway. But no, it makes sense now. I understand.
She got NiH2O4 and
Okay.
H32.
Got it. Got it. Yeah, no, it makes sense. I was okay. Because it still has to be 6 things total, but now it makes sense. It is 6 because there's 5
And
ammonia
then
and 1 H2O. Yeah. Got it. Got it. Okay.
okay. That's cool. Now, wait a second. The next slide report we did was this equilibrium.
Okay.
We can just take a look through. Just tell me when to scroll down and whatever, and I'll set it up. And then we can just go through the
Oh, I
results
mean,
together,
like,
guys.
yeah, start with like what exactly like is your question? Are these your questions or what?
Yeah, like you see all the, like, what do you call it? Oh my God. These questions, like K forward, K reverse.
Okay.
Yeah. So just let me find my pre-labs,
All right.
and then I'm going to combine them together.
Yeah, I'll be right back. Okay.
Oh. there. That. Oh, here you can read the introduction so you can see what like kind of is going on.
Yeah, I mean,
most of it's pretty much done except the stat. So I guess.
so like what's the first thing we have to do, like in terms of like answering the questions?
Hold on.
Yeah.
So it's like spreadsheet simulation. So let's see, download the Excel file, a Chatelier from the Blackboard site for this course. Okay. Okay, so this is part 2.
Let's see.
Hey. Okay. Yeah. Check you out. Okay. Fine. Okay, so just accept the chapter now. All right, so the experimental you see here and stuff. Experiments 1, 2, and 3 we kind of skipped through, but I still put them in.
Okay.
Just for purposes, I guess. Good amount is done, as always. It's good. No data part because we're not actually in lab. Okay, so we have this Excel sheet here, right?
Yeah.
This is the Excel sheet. Okay. So, so I guess over here it says, okay, print out pages 3 to 6 and record work on those pages. Attach those pages you'll have for you, and I need to include the results of your spreadsheet simulation in your notebook. Not forget the question on page 6, the results of your chemical experiment should be written in your notebook. So do I have a discussion? Okay, so kind of confused as what's asking me to do here. So let's see. Okay, so let me copy and paste. They say to copy and paste these pages. the hell is this?
Yeah, it's probably because you're copying pasting from a PDF.
I should convert it, right?
I mean, they should let you have access to the doc version of that, but yeah, it's still copied more than I thought it would have. Because I was gonna say it wasn't gonna copy like any of it, but it still copied a good deal. So, you know.
How do I convert it? Oh, Google Docs?
Yeah, I suppose you can use stuff like that. I mean, it might see if you can open it using, I mean, it still was, I think it was still able to copy and paste most of it. The things that
but
it
it
wasn't able to
wasn't
copy
the
and paste.
underlines and stuff.
What's that?
And it wasn't really doing much of that, no.
Mm-hmm. Like what was the stuff that it wasn't able to copy?
Like all the lines and then half of the other stuff.
Oh yeah, like the formatting. Yeah, like yeah, you
How
could
do
use like
I
some type of converter. You know, it'll try to do it.
how do I upload a
Imperfect.
file again onto?
You can like drag and drop it.
Oh,
Oh well, yeah. Have open up Drive instead of Docs.
okay.
Just straight up like your Google Drive. Yep, drag and drop it there. Yeah, just throw it in. That's where.
Oh, wait. I got it. So okay.
Yeah, if dragging and dropping doesn't work, you could just hit new and upload it like that new file upload. Then find it in there. It's called Equilibrium PDF.
Yeah, I know. I gotta
Okay.
re-download it, I think.
Show in Finder. It's under downloads, yeah. Recent is just, whatever's recent. Okay.
All right, there we go.
All right. All right, and then yeah, hit that. Then open with Google Docs. Try that. Yeah, it should be like converting it right now. Let's see if it does it. There might be some things that, well, that's like an error. Yeah, some of it is.
Retry again.
There's like so yeah, try that. Yeah, there's like there's like connect more apps or whatever. Maybe there's something that's like a PDF converting app. Try like looking for PDF convert, like in the search apps,
What is this?
in the search thing. Like I don't know if that does it, but you can look like PDF convert, like yeah, PDF converter. Maybe there's a couple of those. You can try them.
PDF to Word converter. I guess.
Yeah, there's going to be like, a couple of them that will try to do it. It's still, you'll probably still see some like imperfection in like the formatting of it. Oh yeah, you already have it. You can just, it's already selected. Yeah,
Oh.
just hit convert. There, now download that. Let's see if that works. Yeah, it's still effed up. Because like, it's
Well, let's
hard
see
to
how
do it.
effed up it is. Actually, it's not that effed up.
Okay.
Yeah, the important stuff is not effed up.
Okay.
So, okay, that's fine.
Yeah.
So I can just literally copy and paste this.
Mm-hmm.
Yeah, awesome.
There
And
you
I
go.
can just I can justify it, right?
Yeah.
Make it look nicer.
Just do a control A. But yeah, you're good.
I did it.
Yeah. Yeah, I'm back, by the way.
Okay. Okay, so